All right, and hello everybody. Welcome to the webinar today. Today's webinar is going to be on using ImageJ and FCS Express Image Cytometry. We're going to give an intro to ImageJ exporting to export your data to FCS Express, importing that data, and analysis for that data within FCS Express. So we're going to tell you first a little bit about some ImageJ macros that the team at Novo Software has created that are going to allow you to take your multi-parametric imaging data, your kind of cell-by-cell data, all of the segmentation and results that you've created from that. We're going to be able to import that directly into FCS Express so that we can start looking at that in a flow cytometry style manner. Again, if you have questions during the webinar, feel free to write them in the questions box. We'll stop maybe about halfway through for some questions, and again at the end for any questions, additional questions that you may have. So first, I wanted to just tell you a little bit about FCS Express and how FCS Express may be able to help you out when you're dealing with your ImageJ data and what we can do for you. So FCS Express has been developed as a powerful flow cytometry data analysis and presentation software, and we've been proud to release the Image Cytometry product in the last year or two. The main principles of flow cytometry and image cytometry overlap, such as taking multi-channel fluorescence data and plotting that data in scatter plots and histograms so that you can look for statistically significant differences and changes. But what we can now do is take your image cytometry data a step further by leveraging the most powerful analytical tools in our flow cytometry analysis software. So you can look at your multi-parametric data sets and relate those results from scatter plots and histograms and dot plots back to the original image for confirmation of your results. And in the end, FCS Express Image Cytometry is going to allow you to spend less time reviewing large, kind of overwhelming data sets or looking at raw microscopy images and just kind of saying this is brighter than that, this is bigger than that. By using tools that have been proven to handle large data sets with millions of data points, and this I'm referring to flow cytometry data, of course, and again, you're going to be able to access all of the raw images whenever you need. So again, today I'm going to go over some of the basic functions in FCS Express. We're going to talk about the new ImageJ macros that we put out as well. But let me introduce you to the workflow for FCS Express Image Cytometry first, so you understand where ImageJ actually fits into this workflow. So for a particular image cytometry experiment, what you're going to be doing is first you're going to be acquiring an image, of course. This can be on a microscope, a confocal. This can be from a digital camera, a super high resolution digital camera. This could even be from something off of your iPhone. It doesn't really matter where that image comes from, but as long as you can segment that image in the second step here, which is something that ImageJ is very good at doing at, what we can do is do image segmentation, meaning that we're going to find cell by cell or object by object data, and we can extract features from that data using ImageJ. Things like area, means, medians of fluorescences, perimeters, shapes, all sorts of form factors like that. When we have this feature extraction, and we have the image segmentation and we have the raw images, what we can then do is export that data in a format that FCS Express can read. And in this case, what we can do is take that image segmentation and feature data, bring that into FCS Express for complex data analysis, for report generation, and for communication and publication of your results. So FCS Express is kind of a full package for you to analyze data and make all these great looking reports and keep things in a way that you could even put these into a lab notebook if needed. So let's talk a little bit more about ImageJ now. And how FCS Express is going to work in relation to ImageJ. Because of course we have to do these first image segmentation and feature extraction steps. So what the team at Novo Software has done is we've created a few ImageJ macros that will help get you on your way for creating this feature extraction or export data. Now on our website at denovosoftware.com we have a manual, and of course we have tutorials available. one of these tutorials that's available is for ImageJ export. And I'd like to start here to start talking about the different macros that we have and what they can accomplish and the situations that they should be used for. So let's first take a look at this overview to discuss what these macros are actually going to do. We have two macros that we've developed. One is essentially the export macro, which is this DNS macro example. And here on this page, again, you can download this macro and you can also get more information about it. And we also have something that's called the DNS Analysis Creator macro. And we're going to talk about this DNS Analysis Creator macro later. And we're going to start right now and talk about the export macro to begin with. So essentially what the DNS macro example or the export macro is going to allow you to do is it's going to allow you to process your images, export that segmentation and single-cell image analysis data and results in a way that FCS Express for Image Cytometry can read. And what's going to happen is that a text file with the object-by-object, or in most cases cell-by-cell numerical results is going to be generated. A mask of the images of the segmented objects is going to be created, and then a summary of the analysis in a format that FCS Express for Image Cytometry can read is going to be created, and that's going to be called an IJ out file. And that's what we're going to be importing directly into FCS Express. And when we have this text file with the numerical results, the mask of the images of the segmented objects, and this IJ out file, FCS Express is going to allow you to link up all of the multi-parametric numerical results with the actual images and spatial information all in one place. So there's a number of situations that this macro can analyze in individual or analyze images. The first example is going to be one image at a time. So say you have one image, it's maybe stained with DAPI for DNA, and that image sits in a folder. This macro can go into that folder and analyze and segment that one image and then create the export file. The other case is that we can work with images that belong to a grouping. And what we mean by a grouping is an image essentially with multiple channels. So most of the time when you're doing a fluorescence microscopy experiment, you're not just imaging DNA, right? You may be having a DAPI stain. You may also have something like a CD4 or some other protein stain in there. We're actually looking at a blue, a green, and a red channel. So we can certainly come in and set up the ImageJ macro to export the data for an image with multiple channels. We can work with images from multiple wells in a multi-plate format. So this means that you can work with 96-well plate-based data. You can work with any size plate that you have. FCS Express and this macro are completely formatable to work with anything, from a single image to 6 well plates to 1,500 well plates. Really, the sky's the limit. It just depends on your computing ability, I guess, how many images you can handle. Now, another case that happens is sometimes you'll have multiple fields of view for an image. For instance, if you have a multi-well plate, and in each well you may have 3 or 4 fields of view for that well. FCS Express and this ImageJ macro that we've put together are going to allow you to analyze all of these fields within one well, combine them and pool them into one data set. And then when you put them or export them into FCS Express, we can reconstruct them in a tiled or montage manner so that you can view that pooled data. Now, another instance of working on multiple fields of view is if you have a large tiled or montage image, such as a tissue or histology section, where you've actually imaged, say, a centimeter's worth of tissue in 20x fields, and now you've ended up with 300 fields. We can certainly process those individual fields in ImageJ. We can then reconstruct all of that data as one large data set in FCS Express and stitch together all of those images and use them together in the data set in FCS Express. And again, the last thing we can do here is we can do any combination of multiple channels, of multiple wells, and multiple fields of view. So any type of permutation of these things can be done. So that being said, essentially you can see that we can work with just about any type of imaging format. Let's open up ImageJ and take a look at what this export macro means. And then we're also going to come back and discuss the second macro that I described, which is this analysis creator macro, which is going to help us analyze some of these large data sets. So I'm going to minimize some of my windows here, and I'm going to open up ImageJ. And give this a moment while it opens up. Okay, so here comes ImageJ. It's on the top of our screen now. And what we have is at our website, you can go here and at www.denovosoftware.com, we have an ImageJ page where you can actually go and download these macros. But we have two macros that we want to load. This DNS macro example, it's an IJM file that we can load, and this macro creator, analysis creator, another IJM file. So let's begin by loading or installing these macros. So I'm going to come into plugins and macros and install, and I'm going to find the location on my desktop where these macros have been placed. The first one I'm going to load up is this analysis creator macro, and I'll click open, and you'll notice that in ImageJ in our panel here that we now have a square box icon, and when I mouse over this you can see it says De Novo Software Analysis Configuration File Creator. So this is going to be for creating some of these analysis configuration files that we're going to come back to. But the other plugin that we want to install here is going to be for the actual DNS macro example or this exporter. So I'm going to load this in here. And you notice that when we mouse over this new icon that's appeared, it's actually called the De Novo Software Exporter. So we're going to use the combination of these two macros to actually start creating some analysis, segmentation, and results that we can bring into FCS Express. So I have a few cases here I want to examine. The first case is going to be what happens if we have a single image and it's within a folder, or we have a series of single images within a folder. Here is what this particular image looks like. It's actually just a DNA DAPI stained image. What we're going to have ImageJ do is do the segmentation on all of these individual nuclei here and put out some multi-parametric results. What we should probably do is take a look at what's going on in the macro first to get an idea of how this works before we do the export. So we're going to open up this macro so we can see what's contained in it. And you can see here that in the top part of this macro in the text field, we actually just have information telling you how to use this macro. This is essentially the same thing that's in our tutorial section. It's just a very in-depth explanation of how this works and all the instances that it can work. Underneath that, we have all of the macro information that is being used by what we have set up internally to export the data from ImageJ in a way that we need it in FCS Express. I'm going to spare you the details of that, and what I want to really show you is the functions at the end. And it says the functions below this line are ones that the user can customize. So down here is where we've actually just used many of the default segmentation options in ImageJ to get you started. The first thing we're going to do is we're going to open up that image that I showed you. We're going to set an automatic threshold on that. The next thing we're going to do is say which columns of the results do we want to export for our measurements in FCS Express. In this case, it's things like area, mean, standard modal, the min, centroid, just some of the basic statistics that ImageJ can export. We're also going to say, in case that we're working with multiple columns or multiple images, say images other than the mask, if we're masking on DAPI and we have a FTSE channel, we're going to say what columns we want to export from there as well. And again, essentially the same things as up here. And the final thing that we have in this very basic part of the macro is the perform analysis, analyze particles function in ImageJ, which essentially is going to do the segmentation for us. And in the image that we've chosen, it's actually very easy to segment. But what you can do here is if you have any sort of image pre-processing steps, if you have any sort of macro that you want to include in addition to this macro, and maybe just make the functions above this line the end part of that macro, you can simply just record that macro, paste the macro items into here just like you would when you create any other ImageJ macro. And as long as these functions run at the end of the macro, you will be able to export that data to FCS Express in a way that we can read. So let's go ahead and show you how this is going to work. So the first case is we're going to again look at this single image. We have one image and it's within a folder. Again, it's very easy to segment. But what we're going to do is choose this Novo software exporter, and we have two options. One is to select the source of the data to process. One way is that we can actually use an analysis configuration file, an IJX file, and that's what this other little button is going to do for us, but we'll come back to that. In this case, we're just going to say, let's choose the data from a folder. We'll click OK. What's going to happen is our folder is going to open up. We're going to navigate to where that data is stored. In this case, it's within the single image folder here. And we click select. And if everybody can pay attention, this is going to happen very quickly. ImageJ is going to do the segmentation on this image. It's going to pop up. All the results are going to be calculated, and then the macro is going to close itself. And what is also going to happen is within this folder, you're going to notice that new files are going to be created. So I'm going to click select, and it's going to run itself. So you notice that the RI manager opens up, all the results tables open up, everything closes, and now we have a new IJO file and a new folder within our folder structure here. So again, this IJOut file is what's going to be used by FCS Express to load that up in FCS Express. And this second folder here contains our results text file and the masking file. So when we're ready to import this data into FCS Express, I'm going to show you how this works real quickly and how easy it is. All we do is we open up FCS Express, and again, if I'm going quickly for you here, we're going to come back and talk more about FCS Express in a little bit. But what I'm going to do is say insert a dot plot for this particular data file that we just created in our single image run folder. And you notice that we have files of type for ImageJ files, and we can choose this IJOut file. And when we click open, we now have a multi-parametric scatter plot representing all the different parameters for that one particular image that we inserted or that we analyzed. Here's all the parameters that we used or that we measured in ImageJ. We can essentially switch back and forth between all the different parameters if we'd like, and we'll get more into that as we explore what we can do with FCS Express. But I also mentioned a second way to work with ImageJ is to look at two channels or multiple channels. It doesn't really matter if you have two or if you have three or if you have somehow you've done 12 color imaging. In this particular experiment, we have a DNA image, and also within this folder, we have the cytoplasmic stain associated with that DNA image. So cell-by-cell data for DAPI staining and a protein stain. So what we need to do for this particular file, or these particular images, is that we need to tell ImageJ where these images lie, how they're in relation to each other, because we need to know that one's one channel and one's another channel of the same image. And we can do this by using the IJX creator, or this Novo software analysis configuration file creator. So what we do is we will click on this icon, and what it will ask us is where is the directory that contains the image files that we want to analyze. So in this case, we have images in this 2-channel run folder. We click select, and then it asks us select a directory to save the resulting analysis configuration file. So we'll choose the same folder, and when I click select here, you'll notice that an IJX file has been created for this particular experiment. Now this is essentially a CSV file that can be opened up in ImageJ. That means it can also be opened up outside of ImageJ. If we open this up in Notepad, you can see what is actually contained in this file. And let me make this a little bit nicer in size here. And what we want, or what we have in this file, is we have information associated with these images. We know that by default we've inserted what the file name is, how many channels there are. If this was a plate-based experiment, there would be information for the wells. If there's multiple fields of view, you would want to have these field of view row and column in here. But we have on the second lines is the actual locations of these images. So this is going to tell ImageJ where to go to process these images. And all we have to do is just fill in some information about the channel names. So if I know that I don't want any of this well ID, field of view, column information, we can actually delete this since we're working with single images. But in this case, I know that channel 1 is a FTSE image, channel 2 is going to be a DAPI image. And again, since we have no plate information, we're just going to remove this from the formatting, and we're going to click Save. So what this essentially is going to tell ImageJ is that we have the file name and we have a channel associated with this IJX file. And when we click on the de novo software exporter now, we can choose the option for the data source to process is from an analysis configuration file. I'll click OK, and we can choose this analysis configuration file that we just created. Click Open, and what we'll be prompted for now is we know that there's multiple channels because we actually typed these in, if you remember, in that IJX file. If you had 5 or 6 channels worth of information, they would all be located here. And we want to use the DAPI image to actually create the mask. So we'll choose that channel, we'll click OK, and again this is going to happen very quickly. ImageJ is going to open up, it's going to do a very fast segmentation and export of the results, and it's going to put all that information within this open folder. So I'll click OK. Here goes the segmentation, the ROI manager, the data tables, and we calculated the results for not just one image but both. And if we look in this folder, we actually have 2 results files now because we have one for channel 1 and one for channel 2. So let's again pop this data into FCS Express quickly so you get an idea of what that looked like. We're going to again open up a new layout, and in this new layout again we'll insert a plot, and in this plot we'll choose an ImageJ IJOut file, and we'll choose this 2-channel run that we just created and choose the new IJOut file. Now if you remember in the last plot that I showed you, which is on the top of the screen, we only have information for area because we only have one parameter. So there is no discriminator between all of these different results because they're all from the same parameter. But what we have now in our 2-channel experiment is that we have results for a DAPI channel. You can see here it says DAPI mean and DAPI area. But if we were to change here the second one, we come over here, we can choose the FTSE channel. So we can actually start comparing the amount of DAPI intensity in that image to the amount of FTSE intensity in the image on one axis versus another. So this is essentially how we have been analyzing flow cytometry data for the last 20 years, and now we're able to actually view this data in FCS Express that comes from an image itself. So before we go on with FCS Express, I want to show you one last case study of how these macros can work in ImageJ for something a little bit more complex. You can see here that I actually have a plate-based run, or a plate-based assay. And in this plate-based assay, we have 96 wells. You notice that there's a lot of images here, and there's a channel 1 and a channel 2 for each individual well. So what we've had to do in this case is we've used the analysis configuration file creator, and I've already created an IJX file for this. And when you open this up, this may look a little bit overwhelming, but we have something for a file name, a channel, and a well. We know that we have these three pieces of information that we want to put together. And for each set, we have channel 1 and channel 2. We've named each image a channel 1 and a channel 2, and we've indicated which well that came from. Now you may see this and you may think, oh, he had to type these all in manually last time, and we don't want to have to do that for our 96 images times 2 channels. There's a much easier way to work with this data to create these ImageJ IJX files. Simply the easiest way to do this that I found is to actually just open these up in Microsoft Excel. So if I drag this IJX file into Excel, you can see that we are looking at this data now in Excel, and you can start using some of the features in Excel to extract information from this IJX file. So when this IJX file was originally created, it only had information on the file name location. What you can actually do is set up some formulas in Excel to essentially do the equivalent of a regular expression to pull out the channel name from this text, and then you could put that in a column here named channel, and then you could also have a column here named well. And essentially in Excel, that type of regular expression analysis again is going to, when you create this, it's going to say channel 1 in this column, and it's going to say, well, A01 in this column, because we're going to pull that information from here. When you create these formulas, as everybody knows, you can just drag these down through here in Excel, and you'll actually create the IJX file based on these kind of regular expressions in Excel. So there's probably other ways that you could do this. If you have more programming prowess and you know that you can do this in ImageJ through another macro, you could certainly try that as well. But just setting up some simple regular expressions or some simple formulas in Excel will get you through this process very quickly. And if you need to create these in the future for another plate-based experiment, you can essentially just have those formulas in there and just paste the information into this first column. But once you do that, you can save this IJX file in Excel. We actually have this saved already just in the interest of time. But you'll notice that we have this IJX file, and if we were to run the analysis configuration file, it would prompt us to choose channel 1 or channel 2. And I don't actually want to run all these 96 images right now. That would take ImageJ maybe a minute or two to actually do. But what I've done is I've actually run all of these already. And what we get, again, is an IJ out file that summarizes everything we've done in here. We have our 96 images that we've analyzed. And now in our plate export folder, you can see that we have a results file for every well and every channel associated with this experiment. We also have masks associated with every image in this experiment. So this means that if we come into FCS Express one last time here, and we say insert a new layout, what we can do is use the heat map option in FCS Express to start displaying this plate-based imaging data quickly and easily. So I would choose the heat map option here, and I'm going to navigate to where that plate-based run is and that IJ out file and click open. And what FCS Express is going to be doing in the background now, this may just take a few seconds to load, is because we're loading up all 96 images for 2 channels, so 2 times 96. We're also loading up all of the mask or background images for that. And then we're loading in the cell-by-cell or results or multi-parametric results for all of these different channels in this assay. That way we can view, again, all of this within a heat map. We can start using that heat map to find interesting populations or interesting hits in our experiment if we're doing a sort of hit-based experiment. Or even if you're just looking at things like cells that have been transfected in a particular experiment. We can certainly do that as well. But what we'll get is a heat map. Again, if we click on the axis, we can choose any of the different parameters. So if we wanted to look at the channel 2 mean, we can see all the different wells and the heat associated or the intensity of that channel 2 mean fluorescence. And again, if we were to insert something like a dot plot, we can view all of the cells from this entire heat map on one dot plot, and we can view any parameter against any different parameter. So essentially that is how the macros in ImageJ are going to be working. Again, you have two options. One is to just run this exporter from a folder if you only have a few images in that folder, or if you have a more complex grouping of images, you can certainly run the ImageJ analysis configuration file creator in FCS Express to tell ImageJ how we want to link up all those images for use in FCS Express. So with all that being said, I wanted to take a quick break, and I wanted to ask if anybody has any questions about what they've seen in ImageJ thus far, about the ImageJ modules or macros that we've introduced. And if not, we'll go right on to using this data in FCS Express. So it looks like at this time we don't have any questions just yet. Feel free to at any time hop in and ask us a question if you have any. But what I'd like to now show you is what we can do with this data that you've generated in FCS Express and how you can use it to start further analyzing and getting more information about your multi-parametric data sets. So let's start from scratch. I'm going to close one of these layouts, and we'll open up another one here. Give me a moment while I find what we need. And let's talk a little bit about FCS Express first. So we are actually in FCS Express. You may notice that the format that we're in right now looks very much like Microsoft PowerPoint, very much like Microsoft Word, or Microsoft Office in general. The team at Novo Software has done this very kind of shamelessly. We like everything to look like Microsoft PowerPoint or Office. That way you don't need much experience to come in and start using the software itself. The reason is, in Microsoft PowerPoint, essentially you have a blank piece of paper or a blank slide, or in Word you have a blank slide like you see here. If you were to come to the Insert tab in PowerPoint, you'd be able to insert things like shapes, text boxes, and pictures. And the same thing is going to go in FCS Express, except that we can start inserting other objects or other items, things such as histograms, dot plots, density plots, color dot plots, something that we call a picture plot, and heat maps. And these are things that are going to allow you, or plot types that are going to allow you to work interactively with your data, just like you would work with a picture in PowerPoint. So let's start working with the two-color experiment that we ran in ImageJ. And let's show you how that works in FCS Express. So if we'd like to insert a plot, all we have to do is click on the plot type that we want, and in this case I'll choose color.plot, and we'll draw a box on the page just like you would draw an object in PowerPoint, release the mouse button, and then we navigate to where our data is stored. So in PowerPoint you might be choosing an image. In FCS Express, what we want to do is choose from our files of type drop-down. If we're working with ImageJ files, we choose this ImageJ files.ij out. We would choose that file that we want to add and click Open. So once we've done that, we've inserted a plot in FCS Express. This is a two-dimensional scatter plot, essentially, that you're going to be able to look at any parameter that you've collected or measured in ImageJ against any other. And the way that you can change between different parameters on these plots is by clicking and holding on the axis. You can notice I have the choice to choose anything I'd like here. So if I'd like to put area on the y-axis, and maybe we'll put the DAPI mean on the x here, we're now looking at a two-dimensional plot of area versus mean of the DAPI intensity. Now, one thing to mention is that anything about these plots can be formatted. I mention this because I don't want to work with such small, tiny dots right now. I'm going to zoom in a little bit, and I'm actually going to right-click and format this plot. And right-clicking and formatting is going to get you to our formatting window, where you can make any type of changes to this plot directly in FCS Express. So I'm going to change my dot size to 10. What that's going to do is make my dots a lot bigger and easier to view. Now, when I'm working with two-dimensional plots, again, this is very similar to working with a histogram, but you're working with a histogram essentially that has an X and a Y axis. The reason I mention that is if I insert a histogram for this data, and I look at something like the DAPI mean, or again, let's look at the area in this case. This is probably something that you're used to looking at in the imaging world. And you would say if you wanted to find cells that fall above a certain area limit, you would set what you call a threshold. And we can do something very similar in flow cytometry, or in this case with image cytometry, by going up here to our gating and say create a marker. And what we do is we draw a marker and we say we're interested in this region, and we have a statistic that pops up that tells us how many cells fall within this. Essentially, it's a threshold. Now, in FCS Express, this is called a marker. In the imaging world, again, this is called the threshold. But what we can do is set something that we call a gate in the flow cytometry community. And a gate is essentially a two-dimensional threshold that's going to work on an area or on a plot in the X and the Y dimensions. So if I choose gating and I choose something like a rectangular gate, I can draw what we call a gate on this plot, and you'll notice that it's a square box. And I know that in this box I just created here, it's in the upper right, that these cells are going to be large because their area is large, and they're going to be bright because their mean intensity of the DAPI is actually quite high. And when I do this, a box is created here, or a gate, and again this is like a two-dimensional threshold. It's working in the x-axis and it's working in the y-axis. And what we get is instantly some statistics. These can be formatted to whatever you want about how many cells or what's going on with these cells in this particular gate. The other thing you'll notice is that all the cells that appear within this gate are colored the color of the gate, which is going to help you draw your attention to these cells. And it's also going to give us some other information that I'm going to introduce you to in just a few minutes here. But that's how gates can be created. You can create these very quickly and easily. And the other nice thing about gates is that we can do what we call applying a gate, or making a gate associated with one particular type of plot or another. So if we wanted to apply this gate, there's a few ways we can do it. One way is we can insert what we call a gate view. And this gate view is going to show us all the different gates that we created. You're not limited to just creating one at a time. But if I only wanted to look at the cells within this large and bright cells gate on the scatter plot, I can just drag and drop that gate here. And now we're only looking at the cells that apply, that are in this gate. You may ask yourself, what is the power of that? The main power of this is that we can actually then change parameters on this plot, and we know that all the cells that are going to be appearing in this plot are large and bright. So if we wanted to maybe change our y-axis parameter here to FTSE median, when we have FTSE median versus the DAPI mean now, we know that all the cells that appear on this plot are large and bright for the DAPI channel, and we can see how they relate in almost a third dimension to the FTSE median of these cells as well. And what we can do here is insert, say, another gate, and if we are only interested in FTSE bright cells, we can call these FTSE bright cells, and we'll make this color green because they're FTSE, and click OK. Now we know how many FTSE bright cells are included in this particular analysis as well. Now you notice when I created this FTSE bright cells gate that the gate was created over here on the left as well. And we have a bracket that's showing that these gates are on the same level of each other. And what we can do is, if we want to only look at the FTSE bright cells that are also large and bright in the DNA channel, is we can drag this FTSE bright cells gate on top of the large and bright cells. And now the cells that fall within this FTSE bright cells gate are only the cells that are large, that are bright in the DNA channel, and they're bright in the FTSE channel. We can apply this gate then to any other plot in the software. So if we wanted to say look at only the large, bright, and FTSE bright cells in this histogram down here, we can certainly just drag and drop that gate. And now we have the information on the DAPB area of these cells that are large, bright, and FTSE positive. So you can use these combinations of gates over and over and over again to really start zoning in on phenotypes and subpopulations for analysis. But what we really want to do is not just look at these abstract dots or bins in a histogram. We actually want to see what's going on with the image itself, right? So to do that, we can insert what we call a picture plot in FCS Express. And now a picture plot is not simply a raw image or a TIFF or a PNG file or something like that. We insert a picture plot, and a picture plot is fully interactive, and it's going to show us anything or any gates that we've created in our analysis. We also have access to all the channels, so if I said that I have the DAPI and the FTSE channel here, we can simply click back and forth between these two channels. If you had 5 channels, there would be 5 things listed here. And what you'll notice is that there's some cells appearing in red and there's some cells appearing in green. And what this relates to is the cells that are red fall within this large and bright cells gate that we created, and the cells that are green fall within this FTSE bright cells gate. And actually, since this FTSE bright cells gate is a child of the large and bright cells, these green cells represent cells that are large, bright for DNA, and bright for FTSE all at the same time. The nice thing about this is that as we move this gate and as we update this gate, you can see that on the image we're updating in real time in regard to the changes that we make to the gates. And if I came back here and changed my large and bright cells gate, you can see how the image is updating. Now, I mentioned we can create gates on any type of plots. We created essentially a marker here, a one-dimensional gate on this histogram. What we can do is actually gate directly on the image as well. So if we want to create a gate on this image, we select a gate, any type of gate. I'll choose a polygon. And maybe on this image, I'm only interested in these cells in the middle here. These cells are interesting to me. So I'm going to call them interesting, and I'm going to make them blue. And now all the cells that fall within this interesting gate are going to be highlighted blue to show us which cells these are. You'll notice that a text box appeared that has the percentage that these cells make up of the image itself. So we're getting some statistics about that. But you'll also see over here on the dot plot that we have some blue events appearing. And the blue events that are appearing on the dot plot are the blue events that fall within this interesting cells gate. So in flow cytometry, we call this backgating, where we're able to set a gate on one plot and see where those cells fall on another plot. The same thing applies for the imaging world as well. And what this is also very useful for doing is if you had debris or if you had some sort of artifact on your slide, you can certainly just set this gate to pick everything except that artifact. Another thing that you can do when you create gates is we can create a gate here, is we can say let's exclude some cells that are in a gate. And we can call these artifacts. And if we have that artifacts gate created, we can exclude anything that falls within this artifacts gate from our analysis. But in this case, we don't necessarily want to do that, so I'll delete that. Now what I do want to do in this analysis is I'm interested in the interesting cells that fall on our image, and I want to see how many are large and bright, and how many of those large and bright and interesting cells are FTSE positive as well. So again, I can just drag and drop this gate underneath the interesting cells gate, and now the cells that appear within this image or within this gate are interesting, large, bright, and they're positive for FTSE as well. So again, you can build these up more and more and more to find phenotypes of interest. Now what if we wanted to only view those cells on this image? We actually aren't interested in anything that falls out of this interesting cells gate. What I can do is actually apply this interesting cells gate directly to the image, and what that will do is exclude all the cells that fall from that gate, meaning that we're not going to show the cell cutouts from those. And in this case we have a black background, so one thing we can do to make this look a little bit nicer is actually format the background to be black, and when we do this, the image looks a little bit more reasonable, and we can adjust this gate and see where these cells fall. Now, if we have this gating hierarchy set up, and we really want to zone in here on our FTSE bright cells, we of course can certainly apply this FTSE bright cells gate and now only look at the cells that fall here. Using gates and things in this matter is a great way to find outlying cells. It's a great way to find interesting responders in an experiment. Again, if we wanted to create a gate here just to look at this one cell, find out where that cell is, we'll make this cell pink when we color it in. And if we apply this gate here, we can see where that one clump of pink cells actually exist. So really getting from the population down to the single cell level is very quick and easy to do with FCS Express. That being said, I wanted to show you that we can also insert what we call a data grid. And a data grid is essentially cell-by-cell data. You can see here all the different cells associated with this experiment, all the different parameters associated with it. And if I format this data grid, and maybe we only want to look at the DAPI area, and what we can do is actually see the individual cutouts of the cells or the individual cells associated with that segmentation or that experiment. And what we can do is apply any gate against this data grid. So now we're looking at the interesting cells, and as we adjust this gate, the gate here in our data grid is going to be updating. And again, if we want to get a closer look at the cells within this gate 1, we can certainly just grab this gate from the gate view, put it on the plot, and we can see what that cell looks like. And in this case, it looks like the segmentation didn't do such a good job. It's actually 2 clumps of cells. But again, if we wanted to see these FTSE bright cells, we can certainly just drag and drop them on there and see what's going on with them. So we can move from the population of the whole image down to cell-by-cell data for different parameters, to histograms, all the way down to the individual cells themselves. And what I want to do is clean up this layout a little bit, because in the end, what you really want to do with your data is you want to have it in a form that's going to make sense for yourself, some way that you're going to be able to communicate your results to your colleagues rather quickly and easily. So what I'm going to do is drag this data down here. I'll put the image at the bottom. I'm going to bring my gate view up here so I can see what I did. And what I didn't mention yet, besides these statistics that are appearing on the plots, is that we can get any sort of statistic associated with these layouts or these plots simply by right-clicking on the plot and choosing Show Gate Statistics here. And when I insert this gate statistics window, and we'll move things around and zoom in a little bit so you can see this, we get quick and easy default results for all the information associated with this particular plot. And on this plot is gated on now the interesting cells. And what this is showing me is that there's 132 interesting cells falling within that gate. 73 are large and bright, 17 are FTSE positive, and 1 lies within this outlier gate. We're also getting means for the X and Y values here, and the percentages that those represent of the cells that fall within that interesting cells gate, and the percentages that represent the entire image itself, so besides just the cells within the interesting cells gate. At any time, if we want to choose other statistics, you can right-click and check or end-check anything that you want about these statistics, and you're not limited to just using statistics in a dot plot. You can easily click on a histogram, right-click, and get statistics for that histogram as well. You get things like the low bound, high bound, and the cells that are falling within this marker, peak channels, CVs, any type of data itself. Now there's a lot more options for statistics in FCS Express. We can actually do any sort of custom statistic that you'd like in the software. Any of these statistics that you've seen can also be represented in the software somewhere else by inserting a text box. When we insert a text box, we can give this text box anything in it that we want. We can call this My Results just by typing it in. But the other thing that we can do in this text box is put results from anywhere in the layout into a text box. So if I wanted to choose the percentage, this 55.3%, I can actually just drag that in there and say, put a percentage sign next to it, and we can call these 55.3% large and DNA bright cells. So in this manner, you can build up very nice reports and tables. Again, this text box is going to work just like PowerPoint or Word, where you can insert a table in there. So if you maybe wanted to put your results actually into a table to make this look a little bit better, you can certainly do that. But in the end, the really nice thing about statistics in FCS Express is that these things are going to update in real time. So not only is this 55.3% going to update when we change this gate here, it's going to update in our table, and it's going to update in our custom text box through the use of something that we call tokens. We have a lot of information about using these tokens and about using statistics in FCS Express on our website. I'm going to mention where you can get some more of those resources after the webinar is over today, so you can really build up these reports. But in the interest of time, I'd like to show you what happens when you're ready to communicate and publish these results. All we have to do is come up here to the Save Layout As and PowerPoint, or PDF, or even as a high-quality TIFF or a PNG. We would give this presentation a name, and I'll put this on my desktop, and I'll say, let's open the presentation after we decide to save this presentation. And what's going to happen is PowerPoint is going to be opening up in the background. It's going to be populating the PowerPoint layout or the PowerPoint presentation with everything that's within this layout. You'll notice that we're now in PowerPoint. These are objects you can move around, so if you have a study that you need to show somebody or a group of colleagues, you can certainly just bring your data right here. Again, these are super high quality TIFFs or PNGs or data files, and you can make these as large or as small as you want. So if this data needs to go into a poster, this is your kind of last stop shop for that. You can put everything into your poster from here. And what I didn't show you yet that we're going to save for another webinar is that you can do all of this in a batch iterative process. We never do an experiment where we have one image. We usually have a negative control, a positive control, and then maybe 100 or even 1,000 samples in between. We can use the batch actions in FCS Express to actually do that same save to PowerPoint or to PDF, or even to export certain results to Excel, just by setting up these batch actions and clicking Run. And if you had even 100 or 1,000 images in your experiment and you needed to do this analysis that many times, you would click Run, and the analysis would be quickly and easily done and reported out over all of those samples all at one time. So that's the real power of FCS Express. Besides being able to come in and interact between these scatter plots and histograms and images, you can load in as many data files as you want. Since ImageJ can run in a batch iterative manner, you can quickly segment and process hundreds of images. You can then segment, or you can then analyze and report the data for those hundreds of images within FCS Express itself. In the end, you can really build up some very nice reports. I didn't touch on the heat maps that much, but what I'd like to show you is kind of what you can do at the end of the day when you have these heat maps set up and you have an analysis layout set up. A lot of people, they won't work with one heat map or one plate-based experiment at a time. If you're working in any sort of high content facility where you're actually going to be scanning libraries of drugs or libraries of cells, you may be running 2, 3, 10, some places super high content are running 1,000 plates a day. Again, what you can do is batch iterate all of that analysis and segmentation directly in ImageJ if you'd like. Again, ImageJ is free, so you can do that in a free way. And then once you purchase FCS Express, you can certainly bring that data in here, all of your different plates, and you can create very nice analyses like we've done here. We're actually showing this nuclear translocation assay, how we set up our controls to choose positive and negatives, how we verified the data in each individual well by using cell-by-cell, population-based heat map and individual images. And what we've done at the end is actually set up a chart. In this experiment, it's a titration. So we actually have a regression and a dose response curve that we've created directly in FCS Express. And through the use of the tokens I mentioned earlier, as we update these gates, this dose response curve is going to update for us in real time based on where that gate for the imaging data lies. So there's some very powerful things that you can do all within FCS Express to get to these nice looking results and templates and reports very quickly and easily. So with all that being said, I want to actually, we're kind of coming to the end of the webinar here. So what I'd like to do is just summarize everything that we've seen real quickly. We have these two new macros for ImageJ analysis that can be downloaded from our website. Our website is at www.denovosoftware.com. And if you just type ImageJ up here in the search, you'll actually come up with our ImageJ page, and you'll be able to see FCS Express 4 for ImageJ. This is where you can come and download these macros if you want. We actually have all of our macro databases here. You can actually get onto the ImageJ mailing list. We have a link to download ImageJ. And we also have some applications examples, our tutorials. And with these applications examples, you can open these up and see what we've done to actually create some of this analysis and download these yourself so you can try it on your machine at home. Now in FCS Express, things to remember is that FCS Express is going to leverage all of the powerful and proven tools of flow cytometry analysis with many new features so you can improve your image cytometry analysis. You don't have to just look at an image anymore and say this cell is brighter than that one. You can objectively measure these things in a flow cytometry style manner in FCS Express. And you can do this by moving from the population-based level, using gates down to the single cell level at the click of a button. The new image handling features, you're going to have full interactivity between all the data plots and your images and the cell galleries. Everything is kind of state-of-the-art visualization, plotting, and data analysis. And in the end, FCS Express, again, is going to work just like PowerPoint. You'll notice that we're still within FCS Express, and we've set up some PowerPoint-like slides to make everything easy to use and learn. And along the lines of learning, we have many of these tutorials and sample data sets and an online forum, an excellent customer support system, all of this so that you can get up and running with your analysis in no time. And if you have any questions regarding how any of these things work, you can always contact us at support@denovosoftware.com. We'll typically get back to you within a few hours, 24 hours. You can also get a free trial of FCS Express Image or FCS Express Plus, which combines flow and image cytometry in one package. You can get that at our website at denovosoftware.com. Again, this is in the download FCS Express demo. You can go down here. It's the full version. It includes everything you've seen today and a lot more. And as I mentioned, and you'll notice that you are on this webinar today. It was listed on our main webpage. Next week it will be available in our recorded webinar section. So if yourself or your colleagues or somebody you know may be interested in watching this webinar again, you can come to our recorded webinar section or even visit our videos on our YouTube website at our YouTube channel to come in here and see everything that we've been doing. There are some more recorded webinars that use, say, Cell Profiler for segmentation, some very specific user-based applications examples that we've gone over this week. So lots and lots of resources for the imaging community to use. So at this point, I'd like to open up the floor for questions. I know we actually have gone over a lot of stuff today, more so than just looking at the ImageJ macros and FCS Express. So if you have any questions, find the question and answers box. You can type your questions in there, and we'll be answering these as they come in. And if you have any questions after the webinar, if something comes up that you'd like to ask us, certainly, again, feel free to email us at support@denovosoftware.com. We'd be happy to answer your questions, to talk about the software and how it can be used in your particular applications. So at this point, I don't see any questions coming in, and that's fine. Again, contact us after the webinar if you have more. And at this point, we will be ending the webinar. So thanks again for everybody coming out today. I hope everybody gets a chance to try the ImageJ macros that we've created. They get a free demo download from us, and you can really give it a shot with your actual imaging cytometry data that you have, and I hope this will work for you. So thanks again, everybody, for coming out today, and have a great weekend. Bye-bye.